Hey everyone, so now we're on the week 4 lab. This one is a little bit more intensive. As always, make sure you have the lab manual, the PDF available, that you read through it, and that you also open your lab assistant to fill out. And of course the rubrics are on the bottom because that's how we are doing it. Oh, and apparently I decided not to give you the APA blah blah blah, the APA sample paper, which we will fix in a sec. There we go, now it's there. Okay, so just be warned that one exercise does take 2 days. It's the exercise 1, and it helps if I have the right thing open. And there's going to be 3 exercises in here. As always, if you post your tables, and there are tables for each exercise, you'll get 1 as post. So just put them all in like the reply window. Okay, let's go open the rubric. Okay, so here's the rubric. It's going to be 7 for the exercises, 1 for the other stuff. So the first exercise is 2 points, same for the second. The last one's going to be worth 3. So here we have our petita cells, if you skip below, which again is just a copy and pasting of that beautiful lab assistant you'll be filling out. You don't have to use this one. You can just click on it. I just put it there because it made my life easier. So you'll do initial mass, final mass, the difference, percent of change, and then the class average. Ooh, you have to share this. I forgot. So you'll need to share and get that class average because you know I will complain if it's not there. And I'm going to even put a note that you need a class average in here. Need class average. It makes me so happy. Okay, and then we have your hypothesis. Make sure it's testable, right? And go to, that's going to be a graph. Remember, you can just you can do it by hand, take a picture, and then stick it in here. That's fine. You define osmosis, which probably means that I require a reference. Or was I nice? I was nice. That should not have happened. Okay, well, I would say I would probably have a reference for that. And you know how I am. I always want 3 if possible. References. List your concentrations. What type of tonic were they? Excuse me. The lab manual has really nice pictures on tonic. States, very nice. Molarities, tonics, what's your hypothesis? Was it refuted or not? And what do you think for tonic? Okay, then in exercise 2, we're going to have temperature, time, observations, right? So initial, in the middle, at the end. And then you're going to tell me how does temperature affect the rate of diffusion, the potassium, blah, blah, blah. What would the rate of diffusion be greater in cold water or in hot water, boiling water? And then how does temperature affect it? This is another place, if you're not sure, the manual goes into it, but if you still need another source, go look it up. YouTube is an awesome place. And then diffusion across a membrane. So you've got this sucker in here. So you're going to talk about initial and final. Initial observations, final observations, define diffusion, probably want a reference. You can probably just use the lab manual as your reference. Blah, blah, blah. So you test it. What did you see? And then support. What happened to the indicator? What does that mean? Support your observations. Then we put it somewhere else. What happened? What's the purpose of testing it? How is the cell membrane similar? This is going to be the tricky one. So what type of diffusion do you think you're seeing? Exactly, active or passive. See, I got ahead of myself. And then the same size. Okay, so do you think starch and glucose are the same size? Do you think one's bigger, one's smaller? We did do the macro thing last week, so you might want to go check that or Google it just to make sure. I would kind of be like, here's what I think, and then I would Google it to see what's true, and then it would just make me feel better about the whole process, because that's what the internet is for. Just make sure you use credible sources. And then could the speed be slowed or sped? I would probably consider temperature as for that answer. Okay, so hopefully this helps. And you have fun. The reason, if you're curious for the exercises, I feel like the third one is the scariest, so that's why it got a little bit heavier on the point range, because sometimes fear is a motivating factor. Okay, if you have any questions, PM me, and also send a draft if you have time.